again share. Got it. So let me, okay. Let me find the windows. Right, okay. It would be the PowerPoint, then, right, okay. Multiple. There we go. So I don't know which one we have. So, okay. So, hi, good afternoon, everybody again. Nice to see you right before the weekend. I hope you all got great plans for the coming weekend, but before that, I hope, we'll talk just a little more about the microscopy and especially the kind of science that has been done. Just two very short examples, what kind of science was done using microscopy and how that helps biomedicine. So, but before that, just a few things that you may find helpful. So some of these you have already seen last time, and some of these links will get you to the places where, to the contents that we'll cover today. So you see that there is something about bacteria, and again, we'll come back to the SARS-CoV-2 virus that we briefly mentioned last time, and we'll get into a little more details as to why microscopy is relevant here too in combating the virus, the pandemic. So I hope everybody has gotten a chance. If you have not, well, I have the links in the PowerPoint to see the Nikon Small World photo micrography, if you will, competition. So this is basically a collection of all the winners for the annual competition for nice and eye-catching, and as well as, well, at the same time, scientifically interesting micrographs. So these are pictures taken with various microscopy techniques. So you see, these are really colorful pictures of, in the early days, it was just a cell. Later, it becomes some kind of a tissue, and now more and more, you see whole organisms, like a mini turtle. That 2019, you see a turtle that was imaged. But when you click on these, hopefully that takes us to the right place. Can you guys see the web page? Right. So if you click on this, you will see many additional images. So those are these are all the winners, but the first prize is going to be shown on the cover. So there's a lot of colorful pictures of things like, for example, this one is probably kind of a note. Objects, a lot of people have looked at it, but this one it's kind of a nice, and some other things that you will find interesting. So from big things like whole organisms, whole animal, to smaller things like a cell, right? So you have here, for example, these are actually very colorful pictures. What we're looking at here is it's just two cells. So the top one is actually just dividing. If you see the connections between the two, and the bright orange contrast is the mitochondria. So that's you will find in almost basically every cell, every living cell. But the top two cells are just dividing, so they're not entirely separated yet. So the bottom cell, something happened to it. The two nuclei never separated from each other. And you see that this cyan blob, so that's the nucleus. Usually you will only find one nucleus in each cell, but in this case, you see two nuclei right next to each other. So this cell happens to be also a little bit bigger than usual. Abnormalities like this would often result in cancer and some other diseases. So when you imagine that when the two cells after cell division, or after the cells have replicated everything and they're supposed to separate into two daughter cells, but they fail to do that. So the two remain together permanently, and they have to figure out a way to live on, right? So things like this. So looking at microscopy images can really get us pondering, and think about all this normal as versus abnormal behavior of biological systems. So yeah, I thought it will be interesting to just take you guys back to this Nikon small world. So there's a lot of interesting pictures to look at on this website. So among other things, we'll get you excited about what microscopy can do and what kind of things you would be able to see once you stare at anything under a microscope. So, okay, so let's go back. So last time we talked about microscopes, right? We mentioned the two kinds of microscopes, a light microscope and an electron microscope. So by and large, the way that we look at things using different microscopes are somewhat similar at the basic principle level. So for example, on a light microscope, we have something that we call it a light source, so illuminate the sample, and then the signal is collected by the objectives and viewed either directly on the through a pair of binocular, or the signal can be detected with a digital detector, right? So, and depending on how small of a structure we want to see, we can use different objectives with various magnification ratios. 10x, 20x, 40x, or even 100x. And that can be coupled with binoculars that magnify the image even further. But we also learned that there is a limit to which the magnifications can go. For a light microscope, you roughly can go somewhere between 1,000 to 1,500 times before the image will not, the resolution will not get any better if you keep increasing the magnification because of the behavior of light, or in general behavior of waves, right? Light is a wave. The same thing applies to electrons. When you use electrons to illuminate your sample, you also have this effect, except that the wavelength of an electron beam is way shorter. So instead of a few hundred nanometers, we're talking about a few picometers, right? So that gives electron microscopes a lot better resolution because now you're using something a lot finer to probe the structure. So the big difference between light microscopy microscopes and electron microscopes would be that the resolution, right? The resolution level. But you may be wondering, why do we not always use electron microscopes to see things then? Right? So if the resolution is so much better, why don't we always use electron microscopes? So any clues from the audience? Anything? Maybe. So yeah, just make some wild guesses.
Maybe electron microscopes are expensive? Or...
They are. Yes. Good guess. Other guesses? So if you notice the difference between the two images, if you compare the two micrographs, what else do you see? So besides the resolution, besides the fact that the electron micrograph is a lot clearer, more clear.
You can also see more about like the kind of like 3D, because you have like darker areas and lighter areas. I'm assuming those have to do either with like the molecule structure or with like how close or far they are to like the eyepiece. Basically.
Yes. So you're speaking to another advantage of electron microscopy. So here you have this, although it was essentially a 2D image, but you have a very good sense of 3D relationship across or of the structure itself, right? So because of the shadowing effect, so you're shining electron beam onto the sample, and depending on where they are in 3D, you'll get some shadowing because the electron penetration of electron is limited. So that's another advantage of electron microscopes. So, but I wanted to see if you compare these two micrographs, can get some additional ideas to why electron microscopes are not always the tool of choice for seeing, looking at biological samples. You may notice that, well, on the left, I've told you guys last time was this is bacteria still kind of alive. On the right, the bacteria cells are somewhat dried looking. So the sample is completely dried. And that brings us to this more detailed comparison between these two microscope approaches, right? As Kyla correctly pointed out, electron microscopes are a lot more expensive than a light microscope, which is not listed here because depending on how much budget you have, of course. So a typical electron microscope would range anywhere from $500K, $500,000 to something like a $7 to $8 million, and newer ones may be even more expensive. So the light microscopes would be roughly 2 orders of magnitude cheaper. So it would be about $5K for an average relatively good microscope body, to something like $500,000 or $1 or $2 million if you are talking about a high-end light microscope. So that's one thing. But technically, in terms of the technical specs, apparently light microscopes use visible light as the light source, and electron microscopes use beams of electrons. So that is actually the origin or the basis for a lot of the differences that we'll talk about more. The magnifications that we can achieve with light, because of the diffraction, effectively we can go up to maybe 1,000. But with electrons, you can easily get to 10,000 or even 1,000,000 times magnification. So you can get a lot sharper images, right? And but the disadvantage of electron microscopes is that electrons cannot be focused with a piece of glass lens for good reasons, right? So light can go through glass, but electrons cannot. Electrons can penetrate something just a few microns, maybe, thick, without losing contrast or losing intensity. So focusing of electron beams has to be done using coils of magnets instead of a piece of glass. And because you're dealing with electrons, and electrons oftentimes have to be accelerated. So, and the electrons, if there's air molecules around, the electrons will collide with those. And you, again, will lose brightness of the light source and also the focusing, the sharpness of the focus. So therefore, all of whatever sample that you put in an electron microscope, it would have to be dried. So completely dried, and therefore for biological systems, that means it's also dead. So that gives that's what gives this kind of a dead looking and dried looking of the bacteria in the electron micrograph. So I think by now it's clear that why electron microscopes are not always the tool of choice for seeing biological systems, right? Because if you want to observe something live, or if you want to look at something that's more in the native hydrated state, you probably cannot use an electron microscope for the most part. people are trying to overcome that limitation, but the success has been limited. By contrast, light microscopes can easily accommodate hydrated samples, right? You don't dry, when in the high school biology lab, I think you guys probably have done, some does. I'm just guessing, I'm not familiar. You probably don't need to dry your sample before putting them on the microscope stage, right? Or mounting them into a slide format. And you can even look at living samples. I'm going to give you some example in today's discussion. So that's the big difference between really the light and electron microscopy. And as a matter of fact, until recently, the light microscopes, especially the fluorescence microscopes that we'll be talking about in greater detail, are a lot more commonly used for biological research. So, but when you take a basic light microscope that you would find in most biology lab, sitting on a bench, we look at samples through them all the time. And you put some samples, for example, some cells grown on a substrate, on a dish, petri dish, or some other material. And you look through the binocular, you'd be surprised that there's actually pretty poor contrast. You barely find, you barely see anything when you just look through a monolayer of cells that's grown happily in some nutritious medium. So this is one example that I'm showing here. The bright, so-called bright field image does not give much of contrast. That's because let me ask you. So, what's the most abundant material you find in a cell?
Like the macromolecule or
Good guess.
element?
Yes. So
Water?
there's
Or
Is
it just water? Or
right. So you guys, right. The two things that you guys mentioned were the main components in any living biological system. Macromolecules, right? These are the proteins, DNA, and other things that that supports life. But the most abundant material is actually just water. So most of the intercellular space is filled with water, and what is the material that we're looking at here in outside of the cell? It's also water. So you're seeing water against water. So no wonder you're not going to see a lot of contrast, right? So in bright field, but over the years, there has been ways to enhance this kind of a contrast. So there's maybe something inside the cell that, for example, a fat droplet, the cells use that to store some energy, extra energy, just for cases when extra energy is needed, or for making some hormones out of cholesterol. So there are some droplets of lipids that are inside the cell, and they scatter light more so than water, and you can enhance contrast of those structures. So similar things can be seen a little more clearly if you use tricks such as phase contrast, DIC, dark field, and what have you. You don't need to really understand what these really meant. So these are the best efforts people have made in really to see the cells better under a normal light microscope. So I think Megan just also mentioned there's a lot of macromolecules in cells. As a matter of fact, there's probably on the order of hundreds of thousands of different kinds of macromolecules that you find in a cell. But looking at these images, you probably never figure out where each of those molecules are, right? And where they are, how they perform their functions, right? So there's no limited chemical information. So where's the DNA, right? Where are the proteins? questions like that, it would have to be answered with something that's a little more sophisticated. And that's why people have also, in addition to enhancing the contrast of microscopy images, people have also been working very hard in achieving chemical specific imaging, meaning that if I'm interested in a particular set of molecules, how they work inside of a cell, where they are located, and how do they change over time, depending on what the cells are doing. There's, of course, many different ways of doing that kind of a chemical specific imaging. And among other things, fluorescence microscopy is the one that's used the most. So the fluorescence itself is a photophysical process. So it is the process of a molecule receiving some light, but then upon absorbing the energy from that light, it changes from a so-called ground state to an excited state. So this usually only involves the electrons. So a molecule has a nucleus, and then some electrons orbiting around it. So the electrons, depending on what kind of energy level they set in, they can absorb light and become excited. But they don't stay in that excited state for very long because that's not stable. So almost instantaneously, they would undergo some relaxation and then go back to the ground state, and at the same time emit a photon. So emit some light. So sorry about that. Hey, Ginger. Hey, no, Ginger, come here. Come here. Sorry, she wants to be part of the lecture too, I guess. Hey, Ginger, come. Ginger, come here. Sit. Lie down, huh? Lie down. Good girl. Lie down, stay here, huh? Sorry about that. Okay. So the molecules can absorb some energy from some particular light and then emit light as a result of this complicated process. But in this process, the energy that was absorbed is usually lost. A fraction of it is going to be lost to processes like non-radiative transition. So there's some structural rearrangement within the molecule itself. So we call it, for example, the vibration, the chemical bonds, and all that. You'll learn those when you're in college. But that's details that wouldn't be very relevant today. So what you need to understand is that the light that's emitted at the end of the day always is at a lower energy than the instant light. So being at lower energy, it also is longer wavelengths. So therefore, if you look at the spectrum of light, blue light has higher energy, red light has lower energy and longer wavelength. So that means if you shine blue light on a molecule, the best you can get is probably some yellow or orange light out through this fluorescent state. So, but why is that helpful? So you get a high energy, get some high energy light in and get some lower energy, longer wavelength light out. So that actually turns out to be very helpful for chemical selective imaging. Why is that? Because of the difference between the wavelengths of the instant light and the emitted light. So you can easily add a filter in the light path to filter out. So the instant light is, for example, blue, and that illuminates the sample and generates some fluorescence emission that's at longer wavelengths, which is green. So you obviously don't want the excitation of the light source to get into your detector that overwhelms the image. So what you can do is to add a filter. And so in this case, a filter that sits at 45 degrees and also an additional filter that filters out the excitation light further. So after these two filters, you don't have any excitation light left because this filter reflects short wavelengths and only transmits longer wavelengths. So at the end of this beam path, in front of your detector, you only have the green light, which is the fluorescence signal left. And there's no blue light going into your detector. So you can achieve very high contrast imaging using fluorescence. So again, this is one of the primary reasons why fluorescence microscopy has been most popular in biomedical research, because of this using a simple scheme, you can easily separate out the light source from your signal. And just to make that fluorescence process happen, there's a lot of development involved, hardware, but more importantly, and what's actually driving the growth of fluorescence microscopy has been the development of a full spectrum of dyes. I don't think I need to explain to you guys what are dyes per se, right? So anything that you see with a color is in our everyday life could potentially be a dye molecule. So that absorbs certain visible light and then emit in a different wavelength. So there are now different classes of fluorescent dyes. Some are just organic small molecules. I understand that you guys have good chemistry background and biology, of course. So you probably will appreciate some of the chemical structures that we're drawing here. So one of the dyes that's most commonly used is called Fluorescein. So, I mean, that's probably among the earlier dyes, one of the early dyes developed. So because it has the fluorescence, reminiscent of the word fluorescence in it, right? And if you have a solution of these different dyes sitting in a bottle and shine some purple light onto the bottles and turn off the room lights, this is what you'll be looking at. So the glowing of these fluorescent molecules in solution. And obviously depending on the type of molecules that you have in the solution, the emitted light can be very different. Fluorescein in particular is a green emission dye. So it absorbs purple light or blue light and emits green. So, and some other dyes like rhodamine would emit in orange, reddish color. So the word rhodamine probably has to do with the flower, rhododendron, right? Rhododendron, right? So that's pretty common in Oregon, and probably most of us didn't realize that a big driving force, I would say probably one of the biggest driving forces for fluorescence microscopy actually has its roots in Oregon. So have you guys been to Eugene? I guess most of you have, right? So in Eugene, there used to be a small company called Molecular Probes. So it's a small company that hires a lot of chemists who make different kinds of small molecule dyes. They probably only, I would say, maybe 10% or even less of their dyes that they have made got onto the market ever. But that's already a few hundred dyes, a few hundred different dye molecules. They have a very thick handbook of fluorescent probes that whoever would be doing fluorescence microscopy view that as our Bible. So that handbook has now been discontinued. So we strive, a lot of people strive to try to get a copy of that handbook. But anyways, long story short, that company is still there. It still lives in Eugene. So it's still one of the most prominent sources of fluorescent dyes. And that's part of a bigger company now, but the University of Oregon is actually also been instrumental to the success of that company. So Oregon there's a lot of good things about Oregon, so including science and tech. So this is one of the examples. The molecular probes was the single most important driving force for developing dyes that enabled fluorescence microscopy. And there is another class of fluorescent molecules that we often use, which is the fluorescent, which is actually not organic molecules anymore, small molecules anymore. They're still organic, but these are proteins instead. So they're full length proteins that happen to have a core that mimics the organic dye molecule. And that core can be excited by some light, for example, in this case, blue light, and emit just like an organic fluorophore. So in the early days, when the fluorescent proteins were first isolated, it also started with a green variant. So the green fluorescent protein that was first isolated in jellyfish. So one day some guy just saw that during a diving or underwater expedition, we saw some greenish jellyfish, and he got interested and decided to take some of those and take them back to the lab and do some analysis. So the simplest thing that you can do is to just grind them up so that you extract all of the components in the jellyfish and run them on a gel, so that basically is a technique to separate all the proteins and see what the remaining proteins, what band is the band that gives the fluorescence, the green look, so that then isolate that band that gives you this green light color, and did some further analyses and extracted the green fluorescent protein that actually was fluorescent by itself. So that was the origin of the fluorescent protein, a whole panel of fluorescent proteins, ranging from cyan to green to orange to red and far red. So as colorful as you can get with the small molecule dyes. And the fluorescent proteins, because it's a protein, so that can be genetically linked to whatever protein of interest. Some proteins may be harder to put a dye on, but you can use genetic methods to fuse the fluorescent proteins onto the target gene. So that will give that protein, that target protein, a color, so that now you can track that protein, because now each of those proteins will have a fluorescent protein attached to it, like a light bulb attached to the protein of interest. So, and that really opened up a lot of new ways for us to study protein dynamics and protein function and all that. For that reason, it was given the 2008 Nobel Prize in chemistry. And one of the Nobel laureates was rooted in, he was a professor at UC San Diego. So Roger Chen was actually Chinese American, and is a member of the bigger Chen family who has done many great things. Guess where he was born and raised? You can be brave because the answer is Oregon. So he was Oregonian. So both of these fluorescence, the advancements in fluorescence microscopy really have to do with Oregon. So after Roger Chen has some health issues. So after 2014 or 15, he had a big lab, but he decided to retire and come back to Oregon for the last few years of his life. So he passed away 2019 or 2018. But his legacy really is going to go a long way. The fluorescent protein is something that we use routinely in the lab. So that's still one of the most commonly used ways for tagging proteins. So, as Oregonians, we should be really proud that the contributions the state has made really to this great tool. So, and of course, you have the dyes. This fluorescent molecules, you have to have a way to put them on the molecules of interest, right? So I just told you the fluorescent proteins can be put on your protein of interest by genetic fusion. So you can just put a piece of DNA that encodes your protein of interest, and then another piece of DNA that encodes the fluorescent protein, and just make the two DNA molecules into one. And that whole piece then encodes both protein of interest and the fluorescent protein. So that's a genetic fusion. But in order to put the fluorescent dyes, the small molecule dyes onto your target of interest, it's a little more involving. So usually when you throw dyes onto the cell, it's not going to recognize anything specific because those are small molecules, and the cells would either be water or macromolecules. So in order to highlight specific structures, you need something that helps the fluorophores, the small molecule, to reach the target. And one of the most commonly used ways to do that is to introduce something called an antibody. We all think of antibodies as part of our immune system, right? In fighting infections, fighting debris, unwanted debris and things like that. So, but antibodies have much broader use. So sometimes when we have a target of interest in the cells, like for example, there are structures in the cells called microtubules, so which are long filaments, so highlighted in yellow here, so you can see what they look like. So we know what kind of proteins make this make this filament. So in order to highlight these structures, we just take part of this protein, in this case tubulin, and inject them into an animal. And the animal has its own tubulin, but they're given human tubulin, for example, and their immune system doesn't like it. And their immune system will raise some antibody against it, try to fight it off. But over time, the blood or the plasma of the animal will contain antibodies against human tubulin. So you can have, I don't know if I should talk about here, if there's any animal rights people in this audience, but that's how it is done in the industry nowadays. So you draw blood, you don't necessarily have to kill the animal. Usually you don't. You draw blood from the animal, you can extract antibody from it. And with some purification workflows, you can isolate the antibody specific for the human tubulin. Now you have an antibody that recognizes human tubulin. So when you take a human cell and throw the anti-human tubulin antibody onto them, those antibodies are going to bind to the human tubulins. And over the years, people have already also developed ways to develop antibodies that recognize this so-called primary antibody. So you have a cascade of antibodies, but on the secondary antibody that you add after the primary antibody, you can put a fluorophore through chemical conjugation or some other ways, some other methods. So the light bulb can be put on the antibody. The antibody then recognizes specific target. So this is called immunofluorescence because it's based on the immunorecognition of an antibody against their specific target. So with that method, you can really look at a lot of things in cells, not everything yet, because developing an antibody is quite involving. And for most of our targets, biological targets, we still don't have a good antibody. But there are some very good antibodies. For example, in this picture, what we are looking at, the yellow filaments are called microtubules. So these are filamentous structures that can be thought of as highways in the cell. So these are, you see that all of the microtubules originate from a central point in the cell and has a irregular radial pattern extending out. And when the cell decides that something needs to be transported from the center to the periphery or from the periphery to the center, there's motor proteins like trucks that can move along these filaments and go back and forth and to transport things between the different parts of the cell. So these are microtubules. And there are also other filaments, much finer filaments, which is labeled here in purple, which is called actin. So there are also filamentous structures, but they're more like the residential rows. So these are much finer mesh, and they're responsible usually for transporting cells over a very short range. So from your home to the neighborhood, for example.
Isn't actin the stuff that makes up the cytoskeleton?
Actin is one of the cytoskeleton, and actin is actually one of the main determinants of cell shape as well. You see that there's a layer of actin right underneath the membrane, right? So that's for the most part what gives the cell the shape. So I'm going to talk a little more about actin in the next a few slides. But there is also other components of the cytoskeleton. But I'm going to jump onto some other background before we get back to that point. Okay, so other questions regarding this? Okay, we can talk more when we get back to actin again. So, and with this, I'd like to get into a slightly different topic that has to do with microscopy. It is some bacteria called Listeria Monocytogenes. I don't know if the audience is familiar with Latin. I'm pretty sure I'm not pronouncing it right, but because I'm treating it as English, but it should be Latin. So this is a bacteria that cause food poisoning, and is one of the worst kind because it has a lot of this aftermath that you don't want to deal with. So, so the pathogen is a bacteria. So you see that this electron micrograph of one bacteria cell that has a pretty dense body, but also some flagella, some tails that help them swim around. So these bacteria can swim. And because of its dire consequences, it's especially bad for pregnant women, older adults, and whoever with a somewhat weak immunity, for example, newborns, right? So, so definitely this is not something good. And we still suffer from this because it's actually can grow whenever food is not properly stored or stored for too long. So there was pretty much every year there was like an outbreak of this somewhere in the country. And so and one of the things that people noticed was that these bacteria can infect a host cell, but then quickly propagate into neighboring cells. That's how, and they can, of course, use the nutritions from the host cell and replicate themselves, so make more copies of themselves, and then somehow got into the neighboring cells. And in the early days, people didn't understand how that happened. So when you look at this movie, you probably would be thinking to yourself, I never want this bug to be in any of my cells. So the dark stuff, that's the bacteria that's swimming inside of a host cell. So it's going all over the place, sampling the entire intracellular space. But until it replicated itself and until it has grown to a certain size and strength, it's not ready to leave the cell yet. But once in a while, these things will be able to leave the cell and infect neighboring cells. But one thing got people interested in looking at this movie, this kind of a movie, is that the bacteria is very active in the cell cytoplasm. And while they swim around, there seems to be some kind of a comet tail trailing the bacteria. So let's watch that again. Right, so the bacteria main body is black because it's kind of thick. It does not transmit as much light as the remainder of the cell, of the host cell. But there's always some kind of a hazy tail, like a comet tail, that's trailing the bacteria, right? So people know pretty early on that that's not part of the bacteria itself. And when you put this cell under an electron microscope, you see that this black part is the bacteria. And what's trailing the bacteria is some kind of a filamentous structure. But you don't know what it is because, it's electron micrograph and you're not labeling anything. This is a natural contrast from the sample itself. And if you actually look at how fast these bacteria are moving, you would realize that they actually move at an incredibly fast, incredibly high speed. So the length of each bacteria is around 2 micrometers, 2 microns, and the speed at which they are moving is roughly 1/10 of their body length. So in comparison, a submarine, the Ohio class submarine, with the equivalent length would be moving only half of the speed. So, and the submarine, of course, has a propeller. And you'd be wondering what's propelling the bacteria inside the host cell that makes it really powerful and give it the ability to escape the host cell and then reach the next victim, right? So, and that's where chemically specific labeling, fluorescence microscopy comes in, really in helping to answer this question. So, and looking at this electron micrograph, people suspected that it may be some kind of a cytoskeleton from the host cell that the bacteria cell can hijack. So use it to its own advantage in sampling the inside of the host cell and then eventually give it the momentum to escape the host cell and reach the next cell for infection. And as we were just talking about, there are several kinds of cytoskeleton filaments that could potentially be the candidate that we're looking at, judging from the electron micrograph. So one is called intermediate filaments, and the other one is called microtubules. Microtubules are the biggest or the thickest ones. So these are about 25 nanometers in diameter. So these are the highway for high-speed trafficking of things inside the cell. And then there's the microfilaments, also known as actin. So these are the smallest ones, the thinnest ones. These are only one-fifth of microtubules. They're about 5 nanometers in diameter. And the intermediate filaments are about 15 nanometers in diameter. So that's hence the name intermediate filaments. So, and these were made of different proteins. So in order to differentiate, so which cytoskeleton filament is actually responsible for propelling the bacteria cells, you can do some immunostaining, right? So fluorescence staining and see which one is connected directly with the mitochondria. And that's an experiment that people did in the early 90s. And they discovered that it was the actin that the bacterial cells were using to propel the movement. So this is an example of a cell that's infected by this bacteria, this Listeria bacteria. The infection is so bad that the whole cell is pretty much overwhelmed by the bacteria. The bacteria itself is labeled with this red. This is against a sugar that you only find on this bacterial surface. And the cyan or green or blue, so these are basically the different heights of the same protein. So the color coding depicts the height, the Z direction, axial position of the actin. But all of these highlighted areas, especially the cyan contrast and the green contrast, you see that each of them are actually connected with a bacteria. So the bacteria, most of the bacteria that are moving around would have an actin tail attached to them. And just to make things a little more clear, people have used a technique that we just mentioned. So using green fluorescent protein to label the actin. So they created a fusion protein called GFP actin that highlights the actin in the cells and throw some bacteria on into these mammalian cells and see how, while the bacteria is swimming around, how the actin contrast trails it. So this is an example. I hope this takes us to the right place. So this is a live cell video, fluorescence video of actin that's generated by the bacteria in real time. So you can take a look. So as you follow the actin contrast, you'd be able to see how the bacteria is actually swimming around inside in this cell. So it's actually pretty interesting to look at the way that the bacteria cells are moving around. So you probably noticed two things. One is that the most dense area of or the intensity of actin is actually right at the tail. So in the direction that the bacteria is swimming. But as the bacteria swims to a new position, the original spot where there was a high intensity of actin actually disappeared. So the area right behind the bacteria itself is where you find most of the actin filaments. So that's one thing to notice. So let's go back to the other one. So, because of this propelling effect by the actin, some of these bacteria can eventually find their way out. So pay attention to these two guys. That one swam away. And this one is still trying. So it's incredibly fast, and they seem to be really driven to get out of the cell because their mission is really to infect more cells after all, right? So this is where really fluorescence microscopy and even, be it a dead cell as shown on the left, or be it live cell experiment, is really revealing in dissecting some of the basic mechanisms how bacteria infects the host cell and is able to infect more cells as they go through this dormant period when they're preparing themselves, they're growing and they're replicating themselves, and eventually at the end of the cycle they want to get out, and what kind of forces they can they can utilize to achieve that goal. As a matter of fact, in this case, they're actually using the host material. So a mechanism that's present in the host cell itself to help them achieve that goal. So actin is present in the mammalian cell. It's not something that the bacteria carries on its own, but once the bacteria is inside the mammalian cell, so what it needs is to make a protein called ACT-A. So that is a signal for actin to polymerize right at its butt or at its rear end. And the way that this propelling works is that because actin is a polymer, it can extend itself on this so-called active end. The bacteria just puts this signal on one end of it, and that will induce all of the actin monomers that's present in the host cell to keep adding to the end of it. So this is sort of like laying bricks on top of itself so that you can reach higher, except in this case, this is pushing the bacteria forward. But at the same time, this polymer is actually not stable on the other end. So while new actin is being added right after or right behind the bacteria, actin on the other end is constantly disassembled. So that's what gives this comet appearance of this actin network behind the bacteria. So why does the bacteria want to use this mechanism for propelling? Can you think of a reason why you want actin to be disassembled on the other end while being assembled on the end that propels the bacteria? So why does bacteria just makes a permanent actin filament and pushing it forward?
Like
so what's the advantage of doing this?
If you want to change directions, then you don't have to turn. You can save energy.
Yeah, good point. That's a very good point. And there's probably other reasons as well. So just if you think about it, if I give you a hint that there's a limited supply of actin in cells, and think of actin as a building block.
Is it is it because they break it down so we can reuse it?
Yes. Yes. So very good. If you think about this process, right? So the bacteria definitely want to keep going until they get out of the cell. But if they make a permanent actin structure and that used up all of the available actin, they would have to stop moving. Right. So the best way for them to keep moving is to have a transient structure that at the active front, it pushes the bacteria forward, but the other end, which is now not very useful, the best use of the best fate for the other end is to disassemble so that you can recycle the monomers back into the cytoplasm. So that cycle keeps going and the bacteria can basically move forever. Because at any given time, it only uses a small fraction of all the actin pool available actin. So that's one of the classic examples of how pathogens really hijack the mechanisms that's already present in the host cell to their advantage. And this example is from a bacteria. But the virus that we're dealing with now, and essentially all of the viruses that we're dealing with, they also do the same thing. Not using actin in that sense, but they hijack the mechanisms of the host cells to their advantage. So some simple viruses, for example, they only carry a piece of RNA, right? And once they infect the host cell, they use the host RNA processing machinery to translate them back into DNA. And then the DNA becomes more and more copies of RNA, because there's no most of the biology domain, there's no way to replicate RNA directly. So it has to go back to DNA first and then make more RNA. So once the virus enters the host cell, it uses the host cell's machinery to make more of its RNA, make its RNA into DNA, so that the DNA can now be transcribed into many copies of RNA. Each piece of RNA can then be reassembled into a new virus. So that's, again, using the host mechanism to their advantage. So a lot of imaging work was done in the viral world as well. So, but the example that I'm going to give you today is really on how we fight pathogens based on by using microscopy tools or the microscopes to their extreme. So this is example, you have seen this picture yesterday already, the day before already, right? So these orange particles are the COVID-19 viral particles adhering to the cell membrane right before they enter. And once they are in the cells, they can make more copies of themselves, in part through the process I described to you just now. And obviously, you want to understand how the virus interacts and infects the host cells in order to combat, in order to fight it off, right? So, and one of the tools that has really played a big role in this effort is electron microscopy. So I told you electron microscopes have much better resolution. Among all the tools, the imaging tools that we have today probably has the highest resolution. And so that with electron microscopes, especially the cryo, meaning low temperature, extremely low temperature, electron microscopes can see a lot of details of how individual viral particles look like. So here, what we're looking at is some kind of a virus. This is not COVID-19, but I'm showing it just for an example. Sitting on a EM grid that you can load into an electron microscope. So each of these circles is a virus. So that's about 50 nanometers across. So very small. And even with this kind of a microscope, you don't get a lot of contrast from individual viral particles. So you have some faint view of each virus. It's not a lot of detail. But what people can do, especially in the last roughly 10 years-ish, is that they image tens of thousands or even sometimes millions of copies of the same virus. Because all of the viral particles are more or less the same, they have highly symmetric structures and they look pretty much the same. So then you can analyze the individual images of individual viral particles and do computational analyses, among others, classification and averaging, so that you can group similar images that are similar, meaning that the viral particles are probably orienting in a similar orientation, sitting in a similar orientation on the EM grid, and group all of the images together and do some averaging and computation, so that you can now get a lot more detailed look of the viral particle. And despite the low contrast of this image that I'm showing you here, you can actually get to almost atomic level view of the individual virus, and including their surface proteins. So in this case, I'm showing you this COVID-19 spike protein, which is the protein that sticks out of the viral particle and is responsible for interacting with the so-called receptor molecule on the human cell. So this is green. The green is where it's part of the spike protein. That's what it interacts with the human cell. So you can see that you can actually look at the detailed structure of this spike protein And better yet, you can actually see the spike protein in complex with the so-called the human receptor protein called ACE2. So this is the main protein that's responsible for binding the viral particle and then eventually allowing the virus to enter the host cell. So that part of the mechanisms have been worked out, but you still need the detailed structure of this complex in order to come up with a way to, we call it neutralize, or to disrupt the interaction between the spike protein and this ACE2 receptor. So that's exactly what some people did in the last year or so. They, with amazing speed, because of the power of the microscope that we can use. So again, the concept is that the SARS, the COVID-19 virus would have to go through this transmembrane protein called ACE2, bind this protein first before it can enter the host cell. If we can come up with an anti-spike protein antibody or nanobody, which is a smaller version of the antibody, then we can block the binding between the virus and the human cell. And the virus, despite it still being circulating around, it cannot infect any of our cells anymore. And over time, because the limited stability, biological stability of the virus will just fall apart before it gets a chance to infect the human being, right? So that's the basic concept of neutralizing or antibody for viral infection. But how do you come up with a neutralizing antibody? And of course, you can do a lot of screening, and with the hope that maybe something will come up that will do exactly what we wanted to do here. Or alternatively, what these people have done is that they used electron microscopy to look at the structure of this spike protein. This is part of the spike protein called receptor binding domain, or RBD, and look at how it binds to the receptor using the electron microscope. And once they have the structure, they feed the information to a computer and let the computer compute what kind of structures will fit the interface between the two proteins, the ACE2 and the RBD, exactly as the two proteins would. So that the new protein that you come up with would replace ACE2, so occupy the RBD site, such that the ACE2 is no longer available, the RBD is no longer available for ACE2 to bind. So this is a kind of a competition. So come up with a binder that competes with the virus so that the virus loses the ability to bind to the ACE2 receptor. So, and that really requires that we have a very high and precise structure of the RBD ACE2 or the spike protein ACE2 complex. So these people actually attempted this, starting with this experimental structure, and did some computational work and came up with this so-called Scaffold Library. So this is a whole library of potential binders that will compete with ACE2, and then that's a limited library, and then they can go through just a few molecules and see which molecule works the best. So they have come up with a few tens of molecules that they made, and two of them, they call it LCB1 and LCB3, are predicted to bind to exactly where these two molecules would come together, and presumably these two molecules will be neutralizing the virus or inhibiting the virus from interacting with the human cell, and therefore preventing infection, right? So, and that is exactly what happened. So not all the binders do the same, but out of the binders that they looked at, these two, LCB1 and LCB3, they're the most effective in preventing the virus from infecting the target cell. In this case, it's a monkey cell that people often use in place of the human cell. So you look at here, this is the protein concentration at about a picomolar, which is extremely low concentration. The relative infection efficiency from the 100%, the baseline, dropped to almost 0 just within a few picomolars. A picomolar is 10 to the minus 12 of a molar concentration. So these would be predicted to be very effective as a therapeutic in preventing the COVID-19 infection. So hopefully some of these, I think, are in clinical trials, and there are some other labs who developed this. I know of my actually former advisor. He went back to China. He has a lab there. He developed some neutralizing antibodies as well. So these would do the same as what Trump did when he got contracted by the virus. So I think Trump, as well as the Texas governor, they keep telling people that no need to worry about the virus because it's just flu, and no need to wear masks because it's just flu. But the moment when they got contracted with the virus, they inject themselves with neutralizing antibodies, which 99.9% of us don't have access to. So that's exactly what these are for. So when you are contracted with the virus, these antibodies, neutralizing antibodies, will be able to block the virus from further infecting the host cells. So and now your immune system can kick in and clear the infection that has already happened. But by stopping it at that level, your body will be able to handle it from there. So I hope this is another example that tells you what microscopy or imaging can do in something that's directly relevant to our life in terms of improving medicine. So with that, I think we're at the end of our discussion. I would be happy to take your questions or have more discussions on the things we talked about today. All clear?
I got a question.
Yeah.
The scaffold, whatever molecule you're talking about, that binds to the spike protein on SARS sorry.
COVID-19.
Yeah, COVID-19. Yeah, it's SARS-CoV-2 is the virus.
Mm-hmm.
Would that have any effect on the ACE2 functionality?
It would not, because the binding happens between the antibody and the spike protein. So with that binding in place, ACE2 is not even relevant. So because the virus will not even approach or engage ACE2 anymore.
Oh, okay. I misunderstood one of your slides. I thought you were talking about a specific molecule that would. Yeah, that's
right.
Right. So that's not the antibody. That's just part of the molecule, or.
Right. So let me see if I can annotate this. Yeah.
Yeah.
Right. So
because that just looks like a competitive inhibition or something going on there.
Exactly. Yes. So I'm still looking for the right marker.
Yeah.
Draw. Okay. Right. Let me do that. Sorry. Let me go back to the slide and now do annotates and then draw. Right. Okay. So this part comes from the host cell. And
Yeah.
how do I change color? Right. So this is part of the spike protein from the virus. So the goal is to come up with a binder that will be sitting right here, right in between the two. But if you think about the process, after this binder, one of these, I'm sorry, undo. After one of these binders get to where it needs to be, which is right here, it binds to the RBD, the red part. So the ACE2 molecule will never be seen by this complex anymore.
Okay, so
Because the
it mimics
there's-
the
yeah.
ACE2. It mimics ACE2 then. It's
That's right.
it's-
Yeah.
But you called it an antibody. I don't know if this is that the right word for that, or is it just like a
Right.
competitive
This is
inhibition with a like
right.
a like a
So
polypeptide or something?
polypeptide. So there are different kinds of polypeptides. Not everything can be called an antibody, of course.
Yeah, that's what I was confused about, because you talked about the monoclonal
right,
stuff.
okay.
So
Right.
I thought that you were for some.
Okay.
I got this part confused with the
Ah.
monoclonal part.
Okay, okay.
So
I'm
that was
glad you-
my question was, yeah.
Okay,
That was
I'm
my
glad you
second
brought it
question.
up. So a normal antibody would take a Y shape.
Yeah, exactly. Yeah, it doesn't have
right?
the typical,
So
it's not
this is
nearly
the
as
full-length
large as
antibody, right?
a
So,
typical.
yep,
yeah.
this is the heavy chain, and the shorter ones are the light chain. So this is what a full-length antibody will look like. So the kind of antibody that we are talking about here was taken out of it. So it only is this part, one of these. So we call it a nanobody. So
Okay,
it's a
okay,
right,
I got
it's
you.
a
I got you.
yeah, it's a miniature version of antibody.
Right,
But it has its own folding and properties and all that. So
yeah,
yeah, in that regard, it's more accurate to call it a nanobody instead of an antibody.
okay,
so-
yeah, that was, it was just like a terminology thing was.
That's right. That's right.
confusing
Hence, here I said the concept is a neutralizing antibody
Or nanobody, right.
or nanobody.
Yeah, because I
and-
was expecting to see the typical Y
Mhm.
shape of an antibody
Right.
going on.
Exactly. Good point.
But yeah, nanobody, that's. Yeah, because you don't need the rest of the antibody if you're just looking for competitive
Exactly.
inhibition. You just block the binding site and you're good.
Yes.
Is this scaffolding being used in like patient treatment, or like is it being like developed into a drug in like clinical trials or
Yes.
something?
This scaffold is actually very attractive if you're referring to these. So full-length antibodies are nice because you can get them from nature. You can immunize an animal, and a rabbit, a chicken, or llama, or not llama actually, or a cow or donkey, and get antibody back, and that could be used for therapeutic purposes. But the downside of full-length antibodies is that these are really big molecules. And if the drug needs to cross, for example, the blood-brain barrier, which is what separates the blood circulation and the brain, the internal brain circulation, antibodies cannot do the job. And antibodies, even to get to tumor sites, for example, they have limited efficacy. So because, those molecules move too slowly across barriers. But these miniature antibodies, which we'll call nanobodies, are much smaller, about 1/10 the size. So of a full-length antibody. So if you can engineer something, replace the full-length antibody with the corresponding nanobody, their ability to cross the blood-brain barrier or getting to the disease site within the body is much enhanced. So delivery becomes more straightforward. So a lot of companies are now actually developing these nanobodies for therapeutic purposes. antibodies have been, a great addition to our toolbox for therapeutics, but nanobodies are really catching up. So a lot of attention has been paid to that. And as Andrew was pointing out, the nanobodies are not the only class of polypeptides that being looked at for this purpose. So there are even smaller versions called alpha bodies that also would be used for therapeutic purposes. So a lot of protein engineering there. The last example I showed was also protein engineering. So the lead authors actually are very well known for their efforts in engineering artificial proteins. That's not present in nature, but can really replace the molecules we could previously only get from nature. Okay, very nice. So again, feel free to reach out to me. I think Kyla has my contact, and my contact information should also be in the introduction to the Google Doc, right? Yeah, I'll be happy to talk further on any of the topics. Great. So if no further questions, I wish you guys a good, happy weekend and have fun with the course moving forward. So there'll be additional lectures, I imagine, right, Kyla?
Yeah. This is actually going to be our last session. So.
Okay. Oh, is that right? Oh, okay. Okay. Good. All right. Thanks, everybody.
Thank you.
Yeah.
Thank you so much.
Bye now.
I learned a lot about, like, I really did not know how, like, anything about optics, like, prior to this. So that was really interesting.
I'm glad. Right. So reach out to me if you want to talk more and just have a casual chat. As I said, I had high schoolers in my lab before. Many of them actually, they've gone on to doing, a lot of them doing science and engineering. So you can find their information on our lab website. There's a good list of them actually. So yeah.
Yeah, I was going to say a lot of undergraduates really don't like microscopy until they start doing it. And after a while, they really start falling in love with microscopy.
Yeah.
I was kind of in the same boat. Starting off undergraduate, I really hated microscopy. And then I did a lot of confocal and fluorescent stuff in my graduate program. And then I worked with inverted scopes when I was doing neurophys. And
Oh,
so
nice.
I really fell in love with microscopy. It's an amazing tool. So yeah.
Right. I've seen that trajectory over and over again.
Yeah, exactly. It's
So-
the microscopes suck to microscopes are the most amazing thing in the world.
Yes.
So yeah, it's yeah. Microscopy is awesome, period.
Great. Yeah, I completely agree. A lot of my students would agree too. Once they started doing it, they
Yeah.
cannot live without microscopy, because what you get back is just amazing and always attractive.
Or even just a simple dissecting scope, like if you're
Mhm.
looking at bugs or something, that's still amazing. Just being able to see.
Mhm.
Yeah, so
Yes. Great. Well, thanks everybody again, and I hope we'll cross paths again in the future. Okay. All right. Bye now.
Thank you.
Yep. Of course.